Hello everybody. I hope that your week has been well and that you're ready for today's live stream. We'll be covering the characteristics of bacteria and archaea as well as the diversification of eukaryotes. Alright, let's get started. First thing we'll be covering are what are the main characteristics of bacteria and archaea. First questions asking, based on DNA sequence analysis, 3 major domains of life have been proposed. What are the 3 major domains, and what sets of characteristics place organisms into one domain versus another? So the 3 domains we'll be working with are bacteria, archaea, and eukaryotes. The first one I'll be focusing on is bacteria. And one of the key characteristics are its prokaryote structure. And this is similar to archaea as well. Most bacteria have a circular chromosome. Their cell walls contain peptidoglycan and muramic acid. And just to clarify what muramic acid is that it occurs naturally as the N-acetylmuramic acid in peptidoglycan. So that's just within the structure. All bacterial cells use the same type of RNA polymerase. And bacteria use formylmethionine as their initiator amino acid that starts protein synthesis. So this is not phrased as I want. So it's used for formylmethionine as their initiator amino acid. And also just to clarify on what I always get this wrong, formylmethionine is that a formal group is added to the amino group, and so it's specifically used for that initiation sequence. When that group is added, it kind of gives a clue of, okay, we're starting, and that's found in bacterial organelle genes. Oh yes, and the RNA POL is short for RNA polymerase, so the enzyme RNA polymerase. Sorry, not RNA polymers. I will write that out going forward. Okay, the next one we're going to talk about is archaea. Their cell walls do not contain peptidoglycan or muramic acid. I don't think that this is information you will be responsible for because it's not in the key, but instead of peptidoglycan or muramic acid, archaea have isoprene units, and instead of a bilayer form, a monolayer. This is just other biology stuff I know that I thought was interesting to add in there, but I won't put it on the list because you're not responsible for it. Archaea have a circular chromosome. And they have several types of RNA polymerases found amongst the different Archaea species. So and then Archaea also use methionine as the initiator amino acid instead of formal methionine. Yeah, for sure. So the survey comes in around halfway of the live stream, so it'll be in about 6:30. Okay, awesome. And the last domain, the one that we're most familiar with because we are part of it, is the eukarya. And some of those defining characteristics are the double membrane. Hello and welcome, everybody. Which this also includes a double membrane bound nucleus. And also membrane bound. So it includes a double membrane bound nucleus and membrane bound organelles. Translation goes along with transcription. So transcription is DNA to RNA. Translation is RNA to protein. So I like to think of like a transcript as something that if you're transcribing something, it's similar to the original. So RNA is similar to DNA, but it has its differences. Whereas if you're translating something, for example, if my friend only speaks Germany, oh my gosh, is from Germany and speaks German, but I speak English, then I'll need a translator for different languages, like RNA to protein. So I hope that clarifies translation. And amino acids are used within translation because amino acids are a part of the protein. So when it's reading the RNA, amino acids on top of that tRNA are being linked together. And so that's why they talk about methionine and amino acids as being initiators, because they're directly involved in translation. So you'll see MET as the first amino acid usually. It's called like the start codon. Yeah, of course. Eukaryotes have linear chromosomes as opposed to circular that we're seeing elsewhere. And when they have cell walls, remember that eukaryotes don't always, but cell walls generally contain cellulose or chitin, cellulose of chitin. So walls generally contain cellulose or chitin. Oh my gosh. Awesome. And similarly to Archaea, methionine is the initiator amino acid in translation. Awesome. So this is a comprehensive list that gives you the general overview of the differences between the three domains and can help in the future identifying what organism falls into which domain. On the next page, we're going to be moving into the 6 groups of bacteria and what characteristics are used to place those organisms in each of the groups. The first kind of bacteria that we're going to look at are called Firmicutes. And the characteristics that align with this group of bacteria are that it is gram-positive. Its DNA has low percentage of guanine and cytosine. It is rod or sphere shaped, and I'm going to put that by the gram-positive up top, just in the interest of saving space. Some can fix nitrogen. Some can photosynthesize. Some can perform fermentation. Some perform respiration. And members of this group are also known to be pathogenic and cause anthrax, botulism, tetanus, but also some are more mild and can be used to ferment products like milk. And just right up here I'll put can ferment milk. Alright, the next group is spirocales, and some characteristics with this guy is that it is corkscrew shaped. And has flagella that has an outer sheath to it. They metabolize via fermentation. And some fix nitrogen. Many are anaerobic. And this guy has been known to cause Lyme disease and syphilis. Alright, actinobacteria. This guy has a high percentage of guanine and cytosine. I'm just going to put their abbreviations, G and C within DNA. They are gram-positive and are rod and filamentous shaped. They are heterotrophic and parasitic, and they are also the source of many antibiotics. Remember, heterotrophic just means that they get their nutrients, they cannot make their nutrients to sustain themselves by themselves, so they have to get it from organic compounds elsewhere. And they cause tuberculosis and leprosy. And some are abundant soil decomposers. Alrighty. So moving on to the last three. This group of bacteria is called Chlamydialis. It is spherical shaped and very small. All known species are parasitic and live inside host cells. They cause chlamydia, the STD, or STI, I prefer to say. Most common, it's also the most common cause of blindness, actually, in humans. So not the most fun bacteria to be around as a human. Next we have cyanobacteria. These guys are found either on their own or in colonies. They pretty much dominate aquatic environments. Like, they dominate the mass of aquatic environments, I should say. All are photosynthetic. And some can fix nitrogen. And some live in association with fungi. These guys that do so are called lichens. And they're really cool kinds of cyanobacteria. Alright, so the last type that we're going to be talking about are called Proteobacteria. And this guy has 5 major subgroups. They could have a rod shape, be a sphere shape, or have a spiral shape. They have a wide metabolic diversity. Yes, exactly. That's what that means. So, yeah, just dominate the massive aquatic environment is just that they like are a large part of the aquatic environment's ecosystem. They're found a lot in oceans. So yeah, that could be reframed in saying that that is where they're most plentifully found. That's a really good way of putting that. These guys cause cholera, or cholera, however you'd like to say it, dysentery, gonorrhea. Oh, I don't know how to spell you guys. I know that there was an H somewhere and then I just put it in the wrong place. Ulcers, the other fun, rhea, dia. Oh yeah, and of course, no problem. Always put your questions in the chat. I'm always happy to answer. And they're used to produce vinegar, which is kind of cool. If you like caprese salads, you can thank Proteobacteria. I love caprese salads. And just like a little one more tidbit that I think is cool too, there is one of the genus of Proteobacteria. It's called Rhizobium. And it lives in or on plant roots, and it fixes nitrogen, which is awesome for plants. Yeah, Everyone Died of Dysentery. That's a really good book about the Oregon Trail and some of the things I went through. Fun fact about the Oregon Trail, I know that this is not biology, but they did not walk all the way here. Actually, trains had been invented by then, and so a lot of the travel was flubbed for theatrical purposes. Just fun fact. I'm also a history minor. Okay, so they can cause issues when they infect the human cell rate. Yes. So I think, so Proteobacteria is also just like a wide group of the bacteria. So I think this list is just like some are known to be pathogenic and then some are less harmful and can be used to create things like vinegar that we can ingest. So I think it's just a level of, there are a lot of bacteria, a lot of good bacteria, and a lot of bacteria that could be harmful to us, and those can be in the same group. So I think they are different bacterium that cause illness versus can create vinegar, but they're just classified both as Proteobacteria. Yes, pretty much. So like we have bacteria, which is a prod group, and then these are just 6 other groups to kind of define a little bit further. So it's like going from a view of the world to just continents versus, cities and houses, that sort of thing. Okay, awesome. Yeah, of course. Great. So Now we're going to just move on to the questions. And the answers to these questions really depend just on a large deal of how we're defining what means evolved, and many kinds of comparisons can be made. So the examples I'll be using are just like one of many, and if you come up with others, I'm sure that they're just as great. So A asks, what kind of arguments or evidence would you use to support the idea that bacteria are more highly evolved? And also feel free at any time for these questions to throw your ideas out there. For me, for this one, if we consider organisms that are very closely fit to their environments, adapted to what they can live in best, and we say that that is what means highly evolved, then we could say that many of the species bacteria are more highly evolved than humans. We aren't outside as much as bacteria are. We have environments that we can live in. Some of us, have heating systems, cooling systems, things that can make our space more comfortable, and so there's less variability, whereas bacteria are always in their environment and they're adapted to it. So, to put that into words, so if considering organisms that are more closely fit to their environment, as more highly evolved. I'll put this in italics, or try to. Than many. Bacteria are more evolved than humans. Oh, that's okay. Jump in whenever you get here. That's totally okay. Alright, and B. How would you support the idea that humans are more highly evolved? So, for this one, something I would say, if evolved in this case means major changes in morphology that allows the organisms to live in a wide range of different habitats, rather in the last answer when we said very closely fit to one habitat, then we would say that humans are more highly evolved. So the fact that we are able to create spaces in which we feel comfortable and have those spaces all around the world could also be an argument that we are more highly evolved. And yes, I'm going to answer your question. I'm just going to write this down super quick so I don't write down the words that I am saying out of my mouth. I do that all the time. Wait. Would allow organisms to survive. And yes, I definitely agree with you because there is such a wide variety of bacteria, and many of the microbes that we see in lab is just a fraction of what's actually out there. We haven't even discovered a chunk of the bacteria and microbes and archaea out there. And so these questions are more of a jumping off point for you guys to think. And so a lot of these are very malleable questions. There's a bunch of arguments for both, and a lot of the times they overlap. I think a lot of my personal thing is I think that we're all kind of on the same plane. I think plants, animals, humans, bacteria, archaea. Obviously, we have the communication of language, but plants have a lot of their communication. Bacteria can communicate and respond to their environment. So to argue something is highly evolved is always very complex, and sometimes it's a matter of opinion based on who you're talking to. So I know that is not an answer, but I hope that kind of helps. That, like, yes, it's true. The same answer could be used for bacteria, but this is just a specific example of an isolated event. So it's much larger than just these two questions for sure. Yeah, of course. Alrighty, so given what you know about the origin of life on Earth, what characteristic, you want to look for, sorry. Given what you know about the origin of life on Earth, you want to look for life on other planets. What characteristics of the planet's environment would you look for, and explain your reasoning? In this circumstance, what we know so far is that life can exist in the absence of oxygen. For a lot of Earth's life, there was no oxygen. Oxygen is really, really new to this planet. And there was a lot of life back then, bacteria, archaea, that are anaerobic. So we know that life can exist without oxygen. Oxygen is not a necessity. But to the best of our knowledge, from what we can find, it's very hard for any life to exist in the absence of water. So one of the first tests that I would do is check for the presence of free water, and that's what has been happening in a lot of different planets. Obviously, this is not always true. There's actually studies that there might be kind of a type of archaeal life on Mars, which is super exciting. One of my microbiome lab TAs was telling me about that the other day. But this is just a jumping off point. So look for presence of free water, and this just means free-forming water, like the water, nobody brought the water from another planet and poured it and made a lake. Like, it was already there. I am so bad at spelling, you guys. Water. So this is because from what we know, and this is just in the key. Yes, it is water. Because, yeah, exactly. Without water. Alright, and then what kind or kinds of life would you look for? So, because we're not necessarily looking for a planet that has oxygen, we've already kind of said in the beginning, like, it doesn't need oxygen. A lot of species live anaerobically. The first kind of organism or kinds of life that I would look for are probably similar to those of anaerobic or archaeal species. And the reason why we say this is because we know that a lot of archaea are extremophiles, and so they live in very, very extreme environments and can survive through harsh conditions that we couldn't imagine to survive ourselves. And so it would be most intuitive to look for an organism that can withstand such harsh conditions. Well, you can look for all three domains for sure. I just was using Archaea as an example because it is a lot older than us eukaryotes, so are bacteria, and because they can survive in harsh conditions, it might be easier to find those. But you can look for all of the three domains for sure. Yes, pretty much. I mean, like, there, from what we know in the galaxy that we have searched, no other planet right now has oxygen. It's not the dominant gas form. It's only on Earth that we're finding that. So that's why we're looking specifically for anaerobic species, because there's no oxygen on those other planets. Correct. Awesome. Alrighty, and then the last question. What tests or probes would you use to find the kind of life you proposed in Part A? So going with the example that we're given in the key and that I've been using, so if we're saying that we know life can be in the exist in the absence of oxygen, but we looked for free water as an indication of life, and that this life might be similar to those of anaerobic archaeal species, then the kind of answer to this question would be the assumption that life on other planets, so we have that information, and moving forward, we would have to make the assumption that life on other planets evolved similar to life on Earth to determine what kind of test or probe. And so this assumption is supported by Miller's experiments. These set of experiments, basically what they did was indicated that key organic compounds present on Earth can be produced under reducing atmospheric conditions. So basically the key metabolites of life can be recreated under these more pressurized atmospheric conditions. And so if we're assuming that, then we could look for compounds that we know are common, so things that all life forms pretty much use. If we think about that, in terms of the three domains, we have DNA. All of us do. Bacteria have circular DNA. Archaea have DNA. We have DNA. So if we're assuming that the life we're looking at evolved similar to us, that they have these building blocks to create these fundamental parts of our system, then we could analyze the soil for the presence of polymers such as carbon, that are used in carbohydrates, proteins, lipids, nucleic acids. And so I'll write that down first before moving forward, because I just said a lot of words. And carbon is used for carbs, carbohydrates, proteins, lipids, nucleic acids. And so we're also assuming that the life forms have DNA. So you can also use a radioactive DNA or RNA probe. And so these would be similar to highly conserved genes in archaea and other organisms, and so that could give you a better chance of finding possible homology. So this is a complete hypothetical example of, hey, like, I assume there's archaea. I'm going to go look for archaea. If I want to do some tests, then I would look for common compounds used by all life forms, look for the 4 building blocks of life, and maybe do some radioactive genetic probing just to see how similar those gene lines are to us, and maybe possibly find some homology. So yeah, that was the characteristics of bacteria and archaea. We do have a little bit more to go, as always. I will go until it's 7 and then I'm going to stop, but the keys will all be posted. I saved the diversification of eukaryotes for last just because it is something that is covered the most in your general biology and major biology classes, and so I find it to be most familiar. But of course, if you need any more questions, any more help on that, you can just send me a message through Twitch, and yeah, I can help you. So, diversification of eukaryotes. This activity provides an overview of the early evolution of eukaryotic single-celled organisms. The first eukaryotes appear in the fossil record about 2.1 billion years ago. Current evidence indicates that eukaryote cells arose as a result of endosymbiosis, sometimes multiple endosymbioses, among ancestral prokaryotes. The early eukaryotic cells that resulted underwent great diversification to become the 7 major groups of eukaryotes. See figure 277. This activity is designed to help students begin to understand the vast diversity of the protists, as well as some of the evidence and thought processes that were involved in the development of the endosymbiotic theory. 1. Protists have been called a catch-all group. What does this mean? Explain. Just to define, a protist is any eukaryotic organism that is not animal, plant, or fungus. So we would call this a paraphyletic group. I'm just going to write all. They have a large range in size from macro to microscopic. Range in size. Some algae can actually be like 60 meters long, which is pretty crazy. They have a characteristic and overall form. Yeah, for sure. So this this right up here is the definition of a protist. So it's a paraphyletic group that includes all eukaryotes except animals, plants, and fungi. So that's just the general definition. Yeah, it's a little weird. It's like a very general term. I was researching it, of course, before I start doing these live streams, and so that was just the most regurgitated definition I could find that simplified it. So yeah, so overall in form, they have organelles and other distinctive features. They have nucleus and endomembrane system. They have mitochondria, or just genes that are normally found in mitochondria, and they have a cytoskeleton. So that is kind of the answer to this question. Why they're a catch-all group is just because, you know, they are basically like, we've defined plants, animals, and fungus, but there's a bunch of other eukaryotes that don't fall into those three groups, so we're basically like, it's everything else, it's a protist. If it's not a plant, animal, or fungus, and it is a eukaryote, it's a protist. We don't know, you know? So yeah. There are more than 100,000 recognized species of protists. Even though the organisms in this group are so diverse, we can recognize some common themes in their evolution, including the evolution of complex cell structure, novel genetic recombination strategies, and complex life cycles. In addition, this group as a whole has a great ecological importance. Provide at least 2 examples of protists from chapter 27 that illustrate each theme. So don't worry too much, you don't have to go digging. I have gotten some examples out for you. And I think that we'll try to get through this in the next few tables. So there is a protist called Euglenida. And in its cell structure, it has a light detector, an eye spot, a contractile vacuole, pyrenoids, and chloroplast. You don't have to know like what all of these little things are. These are just examples of protists that have really complex cell structures, and it's just examples that your textbook gives you in some tables. So you don't need to know like what an eye spot is, what a contractile vacuole is. Just understand that they can be very diverse in their structure. Paramecium also have a contractile vacuole. They have an oral groove and cell mouth, which is kind of gross to me. They have a little mouth. It's just so strange. They have micro and macro nuclei and cilia. Yes, let me look that up for you. Okay. So, what Google tells me is that an eye spot, also called a stigma, is a heavily pigmented region in certain one-celled organisms that apparently functions in light reception. So that's probably why they call it an eye spot, is that it can, it can recept light almost like we see, which is kind of cool. Oh, the more you know. Thank you for having me search that. That was really interesting. A novel, yeah, it's like an eye. Dude, that's so weird. Like these little, we don't even think about how these guys, that's why I have struggle with humans thinking that they're better than everyone else, because you learn about these little guys and they have mouths, and they have eyes, and they have all these functions, and you're like, what? That's so cool. Alright, so novel genetic recombination strategies. So, Paramecium is also a cool example of this, where it has, because of its macro and micronuclei, because it has both macro and micronuclei, they actually function in different ways. No, yeah, bacteriophages are crazy. They are like not alive, but I don't know, they're like zombies almost, is what I call them. So the micronuclei in this, the micronuclei in paramecium, I will do a different thing. Function in sexual recombination and reproduction. And then their macronuclei function in growth, maintenance of the cell, and also asexual reproduction. A second example of novel genetic recombination is laminaria. And this guy displays alteration of generations, which just means that it switches between being haploid and diploid. And diploid. Yes, so they have, in paramecium, they have two kinds of nuclei. They have micro and macronuclei, and both have different functions. So micronuclei function in sexual recombination and reproduction, and then the macronuclei is what functions in growth, maintenance, and asexual reproduction. Does that clarify? So yes is my answer to your question. Okay, awesome. Cool, I'm going to try to squeeze this in. We'll do what we can. This one is now asking for examples of complex life cycles. Plasmodium is an example. Oh, still on highlighter. So its life cycle is that it has a two-host life history. History with malaria. So this is what causes malaria. And interestingly enough, it reproduces asexually in humans. And it reproduces sexually in its mosquito host, which is cool. I mean, malaria is not cool. Sorry. I just meant mechanisms. I'm just going to skip to ecological importance because I want to talk about it. Yeah, let's definitely clarify that. I did not mean that. So all the species, I would like to say, basically, pretty much more so in association with cyanobacteria, provide basis for the food webs of all freshwater and marine environments. So photosynthetic. Oh, my pen's freaking out today. For food webs in freshwater and marine. That's just one example. Another example is that there's fungus-like protists that are really good at recycling organic matter, which is great for our ecosystem. And then down here it's asking for, I just think endosymbiosis is really important, so I just want to go over this, and I will try to make my writing brief so I can talk about it. But the endosymbiotic theory is proposed that eukaryotes originated when ancestral prokaryotes, which is most likely in Archaea, engulfed. What is man? Oh, sorry. This is not well written. This is supposed to say marine environments, like fish in the sea. Marine as in sea. Sorry. So eukaryotes. Don't apologize. Do not apologize. My handwriting, I understand how I write, to put it into simple terms. So yeah, endosymbiotic theory proposes that eukaryotes originated when ancestral prokaryotes, most likely archaeal, engulfed formerly free-living prokaryotic cells and enclosed them in vacuoles. And these endosymbionts became major organelles inside the eukaryotes. And so the two eukaryotic organelles that were proposed to have arisen were mitochondria and chloroplasts. I know, it's very, very, very cool. I am very interested in archaeal research. They look so similar to us in terms of the way that they replicate their DNA, and in fact, a lot of the times they have simpler mechanisms of a lot of our own cell processes, which I think are cool. Just because we have almost 1 minute left, I'm going to put in the Google form one more time. If you didn't get a chance to fill that out, make sure you get that extra cred. And just to talk through the evidence behind mitochondria are that it's bounded by two membranes. It contains circular DNA, much like prokaryotes. I'm actually going to write this down. double mem. I'm just going to put mem, like membrane. And then it also has 70S ribosomes. Recall that our ribosomes are 80S, and 70S ribosomes are very similar to bacteria. That's what they have. And some of the intermembrane proteins are similar to prokaryotes rather than eukaryotes. So it basically, the mitochondria basically look like a mini DNA. I'm so sorry. Mini bacteria in our cell, which is really cool. The ribosome number, so a ribosome has two subunits. It has the large and the little subunit. And 1 second. I just want to make sure I get these numbers right. Yes, okay, so bacteria have 2 units in the ribosome. The top ribosome is the 50th subunit, and the bottom is the 30th subunit. And together collectively, this is called 70s. I know that those numbers don't add up. Don't ask me. Ask the biologists who like to be very confusing. And then the 2 subunits in eukaryotes are 60 and 40s. And that's collectively called 80s. So it doesn't really mean anything. This 70s ribosome is just like this type of ribosome, this size of ribosome is seen in bacteria. This size of ribosome is seen in eukaryotes, and that's how we can just tell the difference. It's a lot of it's like nitty-gritty information that's not super imperative, but that's what that means in terms, like when you see ribosome units and you talk more about DNA replication. If you go into upper division biology, they talk about like this guy recruits the small subunit and then the large subunit attaches, and you got this, and that. So that's just where it comes from. But the only thing you need to know is that's what bacteria have. Just to finish it up, pretty much chloroplasts have all the same things that mitochondria do, so you can just rewrite those same 3 points. There were a few more questions on the back, but that will be given to you in your answer key. If there's not any more questions, it is 7 p.m. and we are officially good to go on this week's live stream. So, yeah, anybody have any more questions? Yeah, thank you guys so much for coming. I really appreciate everybody. Yeah, thank you guys. Thank you. You too. Have a great weekend. Enjoy the sun. I think it's still sticking around for a little bit. Have a good night, guys. Bye.